In this video, we will explore the second part of photosynthesis, which is carbon fixation. Sometimes carbon fixation is called the dark reactions or the light-independent reactions, but I don't like either of those terms because I think they're both misleading, so we will simply call it carbon fixation, which is what it is. We will talk initially about C3 photosynthesis, and about the Calvin cycle with a key enzyme called Rubisco. And I hope you get to remember that name, Rubisco, because we owe so much to that enzyme. I figure every literate person should know what Rubisco is. Then we will touch on C4 photosynthesis, which is a mechanism to concentrate carbon dioxide for the Calvin cycle and get around some problems that are inherent with Rubisco. This is a quick overview. In the previous video, we talked about light reactions. And what light reactions do is they produce ATP and NADPH using light energy. And that occurs in the chloroplast and thylakoid membranes or in the photosynthetic membranes of photosynthetic bacteria. Now, in chloroplasts, the ATP and NADPH that are produced by the light reactions are all used within the chloroplast by the Calvin cycle. So again, in chloroplasts, the ATP and NADPH are used within the chloroplast for the Calvin cycle. They are not used by the cell for other purposes. What then happens is that the products of the Calvin cycle are used to make sugar, and then sugar is burned by the plant cell's mitochondria to make ATP to drive all of the cellular work outside the chloroplast. And then the Calvin cycle, by using up the ATP and NADPH, return NADP plus and adenosine diphosphate for further recycling in the light reactions. What I'm going to tell you right now is the story of how Calvin and Benson and their colleague Bassham figured out the pathway now known as the Calvin cycle, which is how all photosynthetic organisms and even some chemosynthetic organisms use to fix inorganic carbon and convert it to organic carbon. So this photo here at the right illustrates their apparatus to investigate carbon fixation. Inside this lollipop, they grew cultures of unicellular algae, and they shone light. So you can think of this glass lollipop culture as kind of like a leaf, because it's kind of flat like a leaf and has a lot of surface area for illumination. Now into this culture, they can bubble in radioactive carbon dioxide for these algal cells to fix and turn into organic carbon. At various time points, they can open the spigot and release some algal cells into this flask of hot ethanol, which immediately stops the reaction, and then they can separate the components or analyze the radioactive components by two-dimensional paper chromatography and autoradiography. What happens in that procedure is that the organic molecules are separated in 2 dimensions on a sheet of chromatography paper. You put a piece of X-ray film. The radioactive compounds in the sheet of chromatography paper expose the X-ray film, and when you develop the X-ray film, you see these black spots, which correspond to the locations of the radioactively labeled organic compounds. Then you can compare the locations of these spots to known spots, to the spots of known compounds, and thus identify them. So when they did this experiment, after 30 seconds of labeling in the presence of radioactive CO2, what they found is that the carbon, all kinds of compounds became labeled. We see some amino acids like glycine and alanine, aspartic acid. Malic acid is a 4-carbon sugar. And here we see sucrose, which is a disaccharide of two 6-carbon sugars. So what we see is that in as little as 30 seconds, the radioactive carbon dioxide has been incorporated into all kinds of molecules. Well, if they went to shorter and shorter times, fewer and fewer molecules became labeled. So here we still see, even at 10 seconds, glucose is labeled. But what we see is that the most intensely labeled spot is phosphoglyceric acid, also known as 3-phosphoglycerate. That's 3-phosphoglycerate, which is the same thing as P-glyceric labeled here. And at even shorter times, 5 seconds, this is pretty much the predominant radioactively labeled spot. Here at 2 minutes, you can see, again, lots of other things being labeled. So based on these results, they deduced that this 3-carbon sugar, 3-phosphoglycerate, was essentially the first stable molecule that they could find that was labeled. And it was labeled at the carboxyl group, which makes sense if you're going to be adding a radioactive carbon dioxide to an organic molecule. They further deduced that it must be a cyclic process, because if you went longer times, then the other carbons of 3-phosphoglycerate became labeled. But this was the big question. You figure that if you're going to get a 3-carbon compound as a result of adding a single molecule of carbon dioxide, then there should be a 2-carbon compound. So, 2 plus 1 equals 3. But there was no 2-carbon compound. They could not identify any 2-carbon compound that could act as an acceptor, that could combine with carbon dioxide to form 3-phosphoglycerate. Now, being good biochemists, they hypothesized that this 3-carbon sugar, 3-phosphoglycerate, would be reduced by the products of light reactions, ATP and NADPH, to another 3-carbon sugar called glyceraldehyde 3-phosphate. And they hypothesized this because these are both intermediates of glycolysis. In glycolysis, glyceraldehyde 3-phosphate is gradually oxidized to 3-phosphoglycerate, and that energy is used to generate ATP by substrate level phosphorylation and NADH. Well, since the light reactions produce ATP and NADPH, and it's consumed by the carbon fixation reaction, it made sense then that we could at least partially reverse glycolysis and use the reducing power NADP to take 3-phosphoglycerate to glyceraldehyde 3-phosphate. So this is a diagram that summarizes their hypothesis that, first of all, it's a cycle. There's some compound X, which is still unidentified, which combines with CO2 to form 3-phosphoglycerate. And and then ATP and NADPH are produced by the light reactions to convert 3-phosphoglycerate to glyceraldehyde 3-phosphate. And what they saw is that 3-phosphoglycerate becomes labeled with carbon radioactive carbon dioxide, in both light and the dark. Okay, so this reaction here, X plus CO2 forms 3-phosphoglycerate, doesn't actually require light. However, to go from 3-phosphoglycerate to glyceraldehyde 3-phosphate requires light because they require the products of the light reactions. So based on this hypothesis, they asked, what would happen if there was no CO2? I mean, there was no CO2 at all. Okay? So if they could simply eliminate CO2, they figured that what would happen is that the cycle, this step would be blocked, and what would then happen is that since this is a cycle, all of these compounds would cycle around until they became compound X, at which point compound X would accumulate because there was no CO2 available to combine with compound X and turn into 3-phosphoglycerate. So basically, all of these molecules are running around in this cycle, and suddenly you put up a roadblock, and all of these molecules pile up behind the roadblock. And we ask, well, what happens? What compound accumulates when we interrupt the cycle in this way? By doing this experiment, they discovered, surprise, surprise, it was not a 2-carbon compound that accumulated. It was a 5-carbon sugar with 2 phosphates. And this 5-carbon sugar is called Ribulose. And since the phosphates are in the 1 and 5 carbons, it's called Ribulose 1,5-bisphosphate. Since that's a real mouthful, we will just call it RuBP. Okay. So this 5-carbon compound combines with a carbon dioxide, and it forms a very transient, highly unstable 6-carbon intermediate, which immediately breaks down to form 2 molecules of 3-phosphoglycerate. And the enzyme that does this is called Rubisco, which is the short version of RuBP carboxylase oxygenase. We will just affectionately call this Rubisco. And if you think about it, this is probably the most abundant enzyme on Earth. Every single atom, essentially, of organic carbon on Earth got here by the action of Rubisco. So if you think about the petagrams of carbon that are fixed worldwide on a global scale every year, that is all due to, or virtually all, due to this one enzyme, Rubisco. So, as I said, all photosynthetic organisms have Rubisco, and many chemosynthetic organisms also use Rubisco for carbon fixation. So here is the Calvin-Benson-Bassham cycle. Those are the 3 Calvin and his 2 colleagues that worked this out. They won the Nobel Prize. For this work. And so here we show the enzyme Rubisco taking ribulose bisphosphate and CO2 as substrates, forming 3-phosphoglycerate. That is phosphorylated to form this 1,3-bisphosphoglycerate, and 1,3-bisphosphoglycerate is reduced to form glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is also formed during glycolysis from fructose 1,6-bisphosphate. So it's the first compound that's formed immediately after splitting glucose into 2. And this is actually a very high energy compound. So glyceraldehyde 3-phosphate is high energy. It can spontaneously, 2 molecules of glyceraldehyde 3-phosphate can spontaneously, meaning it's downhill in terms of free energy, to form a molecule of glucose 6-phosphate. What this figure also shows you then is how the back end of the cycle. And to understand the back end of the cycle, we have to understand then how we can actually liberate or generate a molecule of glyceraldehyde 3-phosphate that then can leave the cycle and do other things in the cell. So in a chloroplast or in a cyanobacterial cell, you can think of lots of molecules of Rubisco. They have many, many copies of Rubisco, and they're all busily fixing carbon dioxide as fast as they can. And so if we just consider 3 parallel reactions, there's 3 molecules of Rubisco, each of which fixes a CO2, so 3 carbon dioxides combined with 3 RuBPs, and as a result they're going to form 6 molecules of 3-phosphoglycerate. These 6 molecules will then form 6 molecules of glyceraldehyde 3-phosphate. Let's say one of these glyceraldehyde 3-phosphates leaves the cycle, and we are then left with, in the chloroplast, 5 molecules of glyceraldehyde 3-phosphate. So these 5 molecules of glyceraldehyde 3-phosphate have 15 carbons. 5 3-carbon sugars give you 15 carbons total. What then happens is a complex series of rearrangements, okay, where you get 3 molecules of Ribulose phosphate. So 3 5-carbon sugars with 1 phosphate each. These are then each phosphorylated using a phosphate from ATP to regenerate 3 molecules of RuBP. Alright, so you don't really have to remember all of this. The point then is for you to understand that if you have enough carbon dioxide being fixed, then some of these intermediates in the Calvin-Benson cycle can leave and still leave enough intermediates that can rearrange themselves and complete the cycle. So here is then a diagram of, in a plant cell or even in a cyanobacterial cell, how the Calvin-Benson cycle fits into the metabolic pathways. Here is our Calvin-Benson cycle. Glyceraldehyde 3-phosphate can leave and either participate in glycolysis or form hexose, a 6-carbon sugar phosphate, and these can combine to form polysaccharides for storage, or cellulose, or those other polysaccharides. Other intermediates, the ribulose monophosphates, those can exit the Calvin-Benson cycle, and they can form the sugar parts for nucleotides, the ribose for nucleotides. 3-phosphoglycerate can also exit the Calvin cycle and be converted to pyruvate or glycerol. Pyruvate can be converted to acetyl-CoA. Those carbons can then enter the citric acid cycle. Alpha-ketoglutarate, an intermediate of the citric acid cycle, can exit and become amino acids for protein synthesis. Acetyl-CoA can also be converted to fatty acids, combining with glycerol to form lipids. So what you see is that carbon coming into the Calvin-Benson cycle can go all over the place in the cell, then can form all the carbon backbones for all 3 types of macromolecules: polysaccharides, nucleic acids, proteins, as well as the lipids for lipid bilayer membranes. So let's go back to Rubisco, the key enzyme in carbon fixation. It's a great enzyme. We literally owe all of our flesh to Rubisco, as well as our clothing, the furniture, anything made of wood, the gasoline that runs our cars. It's all pretty much due to Rubisco. Great enzyme. But it has a problem. It has an alternative function, a dark side. It can actually oxygenate RuBP instead of carboxylating RuBP. This oxygenase activity of Rubisco is confusingly called photorespiration. So I'm not sure that you even want to remember that because it's kind of confusing, and I'll try not to say that on a test or in class. But if you run into the term photorespiration, it actually refers to the oxygenase activity of Rubisco. What is this oxygenase activity? Well, it refers to the fact that oxygen, gaseous molecular oxygen, can compete with carbon dioxide at the active site of Rubisco. And this happens especially at higher temperatures, more than 28 degrees Celsius, and when CO2 concentrations are really low, and the oxygen concentrations have become abnormally high. These conditions exist inside a leaf cell, especially in a hot, dry climate, because in a hot, dry climate, plants have to close the pores on their leaves. So the leaves, the inside of leaves, there's a lot of airspace, and there's gas exchange between the inside of the leaf and the outside air through pores called stomata on the leaf epidermis. But that also allows escape of water vapor. So when the plant needs to conserve water, it has to close its pores, its stomata. But when it closes its pores, it can't exchange gas, oxygen can't escape out into the atmosphere, and it can't bring in fresh carbon dioxide. So inside those leaf spaces with the stomata closed, the carbon dioxide gets used up, oxygen levels accumulate, the temperature is hot, and then this happens. Oxygen competes with carbon dioxide, and we get significant oxygenase activity. Oxygenase activity of Rubisco produces this 2-carbon compound called glycolate. Glycolate goes out of the chloroplast. It becomes oxidized in peroxisomes and is eventually metabolized to release carbon dioxide. But in this process, no ATP is made. So yes, it's oxidizing a carbon compound, but there is no benefit to the cell in terms of ATP production. So this oxygenase activity results in undoing carbon fixation. So this carbon that had this glycolate, and those carbons in glycolate were initially fixed from inorganic carbon by photosynthesis. But now this oxygenase activity is releasing that carbon as inorganic CO2 with no benefit to the cell. So it's a highly negative reaction in terms of the cell's goals. Another thing I want to point out here is that this oxygenase function, the oxygen competing with carbon dioxide, it's not competitive inhibition. Oxygen is an alternative substrate, not an inhibitor. Well, over hundreds of millions of years of evolution, some plants figured out a workaround for this oxygenase activity of Rubisco. And this workaround is called C4 photosynthesis. And you can think about C4 photosynthesis as a mechanism to concentrate carbon dioxide for Rubisco. So C4 plants are called C4 plants because if you repeat Calvin-Benson's experiment, Calvin's experiment of labeling with radioactive carbon dioxide, the first product that's labeled is a 4-carbon compound, hence C4, instead of the 3-carbon compound 3-phosphoglycerate, which is called C3 photosynthesis. C4 plants are some of the better known ones are corn and sugarcane. the crabgrass that comes up on your lawn, they're all C4 plants, and they typically thrive in dry and hot environments. As we said, in dry, hot environments, plants have to close their stomata to limit water vapor loss, but they limit gas exchange. So what C4 plants do is they initially take up carbon dioxide by a different enzyme, not Rubisco, but a different enzyme called PEP carboxylase. PEP stands for phosphoenol pyruvate. We'll just call it PEP. And the enzyme that adds CO2 to PEP is called PEP carboxylase. PEP carboxylase doesn't have Rubisco's problem. It has a very high affinity for carbon dioxide, so even when CO2 levels plummet, get really low inside the leaf, PEP carboxylase can still keep working. Another thing is that PEP carboxylase has no oxygenase activity. It completely ignores oxygen no matter how high the oxygen levels get. Now, C4 plants have a little different leaf anatomy. Their photosynthetic cells are now become differentiated into two different kinds of cells. One type of cell is called a mesophyll cell, and it's pretty much scattered throughout the leaf. A second type of cell is called a bundle sheath cell, and these bundle sheath cells are grouped very tightly around the vascular tissues. The leaf veins are the conductive tissues that conduct water to the leaf and sap out of the leaf down to the roots. And it turns out that these mesophyll cells and the bundle sheath cells in C4 plants have different chloroplasts. So the mesophyll cells use PEP carboxylase and C4 to fix carbon dioxide to form a 4-carbon compound, which is then converted to malate. This 4-carbon malate is transported to the bundle sheath cell, and this malate then is decarboxylated and releases carbon dioxide inside the chloroplast of the bundle sheath cell. The bundle sheath cell is actually a C3 photosynthetic cell because it has Rubisco and the Calvin cycle. So the Rubisco in the bundle sheath cells can use the carbon dioxide that's been delivered by malate to run regular C3 carbon fixation. The product of carbon dioxide release from malate is pyruvate, which then goes back to the mesophyll cells, and inside the mesophyll cells they have to expend phosphate from ATP to phosphorylate pyruvate and regenerate phosphoenol pyruvate, or PEP. Now you'll notice that there is a stage here that uses reducing power from NADPH, but that's recouped inside the bundle sheath cells because in this decarboxylation reaction from malate, the reducing power is recovered. So there is no net loss of reducing power to operate this cycle, but there is a net cost of 1 ATP for every CO2 delivered to the bundle sheath cell, delivered to Rubisco and C3 fixation in the bundle sheath cell. So you should really think about the mesophyll cells and C4 fixation in mesophyll cells as essentially operating a CO2 concentration mechanism, a way to deliver CO2 with efficiency to the bundle sheath cells where the actual permanent carbon fixation occurs. But there is no such thing as a free lunch. There is an energy cost of 1 ATP per carbon dioxide fixed. To summarize then, C4 versus C3 photosynthesis. C4 plants have minimum loss of organic carbon from oxygenase activity. because they're operating this CO2 delivery system for Rubisco. As a result, C4 plants grow better under hot, dry climates. But because there is an energy cost, if you have cool and moist climates, C3 plants grow better. C3 plants are more sensitive to atmospheric CO2 concentrations because they have only Rubisco and they lack that CO2 concentration mechanism. So under today's scenario where atmospheric CO2 levels are significantly increasing, we would expect then that C3 plants would actually begin to fix more carbon than C4 plants, or that the rate of carbon fixation in C3 plants would increase faster than the rate of carbon fixation in C4 plants. Finally, to conclude our unit on energy metabolism, let's look at the balance of photosynthesis and respiration. Here is the equations for photosynthesis and respiration, written with a reversible arrow. So if we go from left to right, where we start with carbon dioxide and water, and make glucose and oxygen, this of course then is what we've just talked about. This is photosynthesis. But the products of photosynthesis are then the reactants for respiration. Now let me put in a couple of qualifiers. This is true for aerobic respiration, and this is oxygenic photosynthesis. Now we know from the geological record that we had to have oxygenic photosynthesis being invented first to produce oxygen, and only after oxygen began to accumulate could we get organisms that could now use oxygen for aerobic respiration. But just like there were alternative forms of respiration using alternative terminal electron acceptors besides oxygen, before aerobic respiration evolved, there are other forms of photosynthesis that did not produce oxygen called anoxygenic photosynthesis. And we will talk about that in class. But for now, oxygenic photosynthesis and aerobic respiration are the predominant forms of photosynthesis and respirations on Earth. And they are responsible for carbon and oxygen cycling, global carbon and oxygen cycle. The balance, as long as they remain in balance, we will have a long-term steady state carbon dioxide and oxygen levels in the atmosphere. Both of these processes, oxygenic photosynthesis and aerobic respiration, feature chemiosmotic ATP synthesis, meaning we have ATP synthesis driven by a proton gradient across the membrane, and ATP synthase in both cases is pretty much the same. They're both carried out in eukaryotic cells. In eukaryotic cells, both processes are carried out in organelles with endosymbiotic origins.